Alexander has spent a night out binge drinking, and we're going to check how that's affected enzyme activity in his liver. We think that the more ethanol there is in his liver, the less catalase activity there will be. For our control trials, we soaked a paper disc in catalase and put it in a reaction chamber, separate of the 10 milliliters of hydrogen peroxide. The setup was then placed into a tub of water and flipped, mixing the reactants to produce oxygen gas, displacing the water in an inverted graduated cylinder. To investigate ethanol's effects, we added varying volumes of ethanol to the catalase discs, using a dropper to create 4 test cases, being 0, 2, 4, and 6 drops of ethanol. The procedure was then completed just like before, with 2 trials for each test case. Factors like the temperature of water, volume of catalase, and volume of hydrogen peroxide were controlled. The data was recorded into this table. This data was then graphed to show the trend by using average values for both trials in all 4 test cases plotted against each other. Finally, we graphed the average rates of change in oxygen volume every 30 seconds for all the test cases, and it is clear that less oxygen was observed to be produced as more ethanol was added. This being said, upon consulting outside sources, we discovered that our hypothesis is harmonious with our data and expected results, but it has an incorrect conclusion. This is because on the molecular level, catalase breaks down hydrogen peroxide in 2 steps. It first bonds to one of hydrogen peroxide's constituent oxygen atoms and releases the remainder of the molecule as water. It then bonds to another hydrogen peroxide and reacts it with the oxygen atom it took from the previous hydrogen peroxide, yielding water and oxygen gas. In the presence of ethanol, however, as illustrated by these diagrams, catalase will perform a reaction that simultaneously decomposes ethanol and hydrogen peroxide while yielding acetaldehyde and 2 water molecules. Therefore, in the presence of ethanol, hydrogen peroxide may be broken down without producing any oxygen gas. In our lab, this would result in a decreased volume of water being displaced in the inverted graduated cylinder by oxygen, and thus perceived, but not actual, decrease in enzyme activity. One systemic error that may have affected our experiment is the result of the long tube used to connect the reaction chamber and the graduated cylinder. The length of this tube meant that oftentimes, the oxygen gas released during hydrogen peroxide's decomposition would form bubbles along the long route to the graduated cylinder, instead of streaming in at a constant rate. Upon a large bubble being disturbed, a large amount of oxygen gas would rise to the top of the cylinder at once, thus disrupting our results' accuracy at different time intervals. A 2-pronged solution to this issue would be to use a shorter tube so that there was a smaller volume of tube for oxygen to form pockets in, and also to tilt the tube upwards so that oxygen gas produced would immediately rise to the graduated cylinder and not congeal in the tube. This experiment highlights the interaction between ethanol and catalase, the enzyme in the liver that decomposes hydrogen peroxide into water and oxygen. In the presence of ethanol, this enzyme uses H2O2 to oxidize ethanol to form acetaldehyde, a toxin, and a carcinogen. This experiment is thus a useful demonstration of the damaging effects alcohol consumption can have on the body. Little alcohol consumption, as seen, will not be too harmful as your body can break down small amounts of ethanol fairly quickly. Increased consumption, however, will take far longer to break down. This large buildup of the toxin and carcinogen acetaldehyde can lead to the inhibition of proteins and enzymes in the liver, causing alcoholic liver disease or even liver cancer. Now that we are better informed about the hazards of alcohol consumption, we can dig deeper into the topic, asking questions such as how much alcohol is too much before severe health effects? Can we disable catalase from producing acetaldehyde? And what are the long-term effects of alcohol consumption on the liver on a molecular scale? The logical extension of this experiment is working on a way to prevent the harmful effects of alcohol consumption. While the simplest solution would be abstaining from alcohol consumption, most people are unlikely to do so. Thus, a follow-up experiment could be finding a substance that acts as a catalyst to break down acetaldehyde. This substance would then help mitigate the toxic effects of acetaldehyde by breaking it down quickly, and could thus help prevent liver disease and cancer. In summary, we learned that when hydrogen peroxide is decomposed with ethanol, it produces acetaldehyde, a toxic compound that causes liver damage, and that will be the principal threat to Alexander's health next time he has a night on the town.